All right, today we're going to cover chapter 13 in your book on blood. We're talking about all the different things that blood are made of and all the different parts and how do we clot, how do we have blood types, all of those types of things. So blood, remember back long ago when we talked about tissues, that blood is actually a connective tissue. And it's really several types of cells in combination with fluid. And this fluid matrix allows the blood to move and serve as a transport medium as it delivers oxygen and removes waste products from tissues. So let's talk about the different blood components first. We have plasma formed elements, and then we have this little layer called a buffy coat in between. And the components don't become apparent like this until they go through a centrifuge. So that's what happens when we take a sample of blood and we actually spin it. And these blood tests allow us to gain insight about disease and disorders. So plasma is the clear extracellular matrix, we call it. It accounts for about 55% of blood. The main component is water, but it also does contain proteins, mainly albumin, nutrients, electrolytes, hormones, and gases. These plasma proteins play a role in blood clotting, the immune system, and regulation of fluid volume. Plasma without the clotting proteins is called serum, so you may hear that term used sometimes as well. The formed elements are erythrocytes, which are our red blood cells, or RBCs, leukocytes, which are our white blood cells, or WBCs, and platelets. They make up about 45% of blood. The RBCs are the heaviest of the formed elements, so they sink to the bottom of the sample when it's spun in the centrifuge. And when we measure something called a hematocrit, we're actually measuring the percentage of red blood cells in a sample of blood. You'll hear hematocrit for a long time, so that's something that you definitely want to know. White blood cells and the platelets form actually that narrow buff-colored band just beneath the plasma called the buffy coat. These cells constitute 1% or less of the blood volume. So most of the blood volume is plasma and red blood cells. Something to take note of is the viscosity of blood, or kind of the stickiness or the thickness of blood. Usually blood is about 5 times thicker than water, but a lot of things are going to affect this. So if someone's really dehydrated, it's going to become more thicker or stickier. And if they're overhydrated or they don't have enough red blood cells, it's going to become kind of more thin, or the viscosity is going to decrease. So let's talk about the formation of blood cells, which is also called hemopoiesis. Okay, so red bone marrow is found in the ends of long bones and in flat, irregular bones, such as the sternum, cranial bones, vertebrae, and pelvis, which we did learn about before. And lymphatic tissue is found in the spleen, lymph nodes, and the thymus gland, which we just talked about last chapter. So the differentiating here is that the red bone marrow produces all types of blood cells, and the lymphatic tissue just produces lymphocytes. Okay, an adult normally produces 400 million platelets, 200 billion RBCs, and 20 billion WBCs every day. So this is happening all the time. If you look on page 246 in your book, you'll see a whole little chart there that talks about all the different names of red blood cells, white blood cells, and platelets. You don't need to know all of these things. You need to know erythrocytes or red blood cells. We will talk a little bit about the different white blood cells, but just a little bit. And then thrombocytes really are platelets. So red blood cells, their job is to deliver oxygen to cells and remove carbon dioxide. So that's their main role. They're very critical to survival because of this job alone. The RBCs are in this disc-like shape, and it provides a large surface area through which oxygen and carbon dioxide can diffuse, so that it allows it to do its job better. Red blood cells lose almost all of their organelles during development because they lack a nucleus and DNA. They really cannot replicate. So we just produce them and they cannot divide. The cytoskeleton of the red blood cell contains stretchable fibers that make it flexible, allowing it to fold and stretch as it squeezes through tiny capillaries and then spring back into shape. A single drop of blood contains about 5 million red blood cells. So you can see how tiny red blood cells really are. Hemoglobin, okay, is part of a red blood cell. So more than a third of the interior of a red blood cell is filled with what we call hemoglobin. It really gives blood its red pigmented color. This is looking at one kind of piece of hemoglobin here. It consists of 4 ribbon-like protein chains called globins. Bound to each globin is an iron-containing molecule called heme. Okay, this is why we need iron, all right, to have good red blood cells in general that are going to do their job. Each heme molecule can combine with 1 molecule of oxygen. Therefore, 1 hemoglobin molecule can unite with 4 molecules of oxygen to form oxyhemoglobin, okay? And that's how we get oxygen to the tissues. Each red blood cell contains 200 to 300 million molecules of hemoglobin. So it's not like there's just 1 molecule of hemoglobin inside a red blood cell. Millions of them, okay? Something that I want to mention to you too, because throughout this, we're going to see pictures of red blood and blue blood. Blood is never blue, okay? It's always red to some degree. I just don't want you to get, confused about that. If you look on page 248, you'll see hematocrit, hemoglobin, and red blood cell counts, okay? I do want you to think about those and to know those. You're going to need to know them going forward. So now is a good time to commit those to memory, okay? These are normal blood values, so you'll be looking at lab values for the rest of your career as a nurse, and you'll need to know these spectrums or these values here. So let's talk about a red blood cell life cycle. Really, red blood cells circulate for about 120 days and then they die, break up, and are consumed by cells in the spleen and the liver. The body has to constantly reproduce red blood cells to maintain homeostasis, and this is through a process called erythropoiesis, okay? This process is maintained through a negative feedback loop, which we just talked about last chapter. So what really happens and what causes red blood cells to be formed? Oxygen levels decline as damaged red blood cells are removed from circulation. The kidneys detect that these oxygen levels are decreasing and secrete a hormone called erythropoietin. Erythropoietin stimulates red bone marrow to create new erythrocytes or new RBCs. Immature erythrocytes are released into circulation. After 1 to 2 days, they become mature. As this number of red blood cells increases, it causes oxygen levels to increase. Therefore, less erythropoietin is produced and red blood cell production declines, which is the negative feedback cycle. So let's watch the animation.
Red blood cells circulate for about 120 days. At that point, they die, break up, and are consumed by phagocytic cells in the spleen and the liver. Every second, about 2.5 million red blood cells are destroyed. Consequently, the body must constantly produce new red blood cells, or erythrocytes, to maintain homeostasis. The process of producing new erythrocytes, called erythropoiesis, is maintained through a negative feedback loop. The process begins when oxygen levels decline, as damaged red blood cells are removed from circulation. The kidneys detect the declining levels of oxygen and respond by secreting a hormone called erythropoietin, or EPO. EPO stimulates the red bone marrow to begin the process of creating new erythrocytes. An immature form of an erythrocyte, called a reticulocyte, is released into circulation. After 1 to 2 days, the reticulocyte becomes a mature erythrocyte. As the number of red blood cells increases, oxygen levels rise. Less EPO is produced, and red blood cell production declines.
So a very typical negative feedback cycle. So what happens after those 120 days and the red blood cells are no longer of good use to us is they're actually broken down into multiple things. So macrophages in the liver and spleen ingest and destroy old red blood cells. In the process, hemoglobin is broken into its 2 components, globin and heme, Okay, globin is broken down into amino acids, which are reused to create energy or create new proteins. Heme is broken down into iron and bilirubin. Okay, the iron is used to create new hemoglobin, so we recycle it. And bilirubin is excreted into the intestines as part of bile. When the destruction of red blood cells becomes excessive, hemolysis we call it, the body cannot get used to these increased amounts of bilirubin. And what happens is this excess bilirubin enters the tissues, causing the skin and sclera to turn a yellowish hue, or jaundice. We learned that term before as well.
As a red blood cell ages, its membrane weakens, becoming fragile. As it passes through the narrow capillaries in the spleen, it begins to break down. Macrophages in the liver and spleen ingest and destroy old red blood cells. As they do, hemoglobin is broken down into its two components of globin and heme. Globin is further broken down into amino acids. The amino acids are used for energy or to create new proteins. Heme is broken down into iron and bilirubin. Iron is transported to the bone marrow, where it is used to create new hemoglobin. Bilirubin is excreted into the intestines as part of bile.
So just a recap of what I just told you. White blood cells, also known as leukocytes, another part of the formed elements. They're really the fewest of the formed elements, but they're still really important because they're our body's line of defense against infectious pathogens. So it's part of our immune system. The body does contain 5 types of white blood cells, all different in size, appearance, abundance, and function. I'll let you know which ones you need to know about. All leukocytes contain a nucleus, which is different from our red blood cells, so it's a differentiating factor. Leukocytes contain other internal structures that look like granules when stained and examined under a microscope. So first, we differentiate them as granulocytes and agranulocytes, meaning with these kind of granules or without these granules. The granulocytes, if you're looking on page 252, 253, you can see what I'm talking about here. So there's 3 types of granulocytes, 2 types of agranulocytes. Okay, so you definitely need to know neutrophils and lymphocytes. Okay, most importantly, you need to know that neutrophils are the most abundant. Okay, and sometimes we look at the amount of neutrophils when we're looking at infectious processes because these are kind of young cells, young white blood cells. And if we have a lot of young white blood cells, it's going to be identifying to us how bad the infectious process is. And then lymphocytes, which are agranulocytes, are the second most numerous. Okay, they're the smallest of the white blood cells, and they're going to play a role in immunity. So when we get to the chapter talking about immunity, we're going to talk more about those types of white blood cells. Platelets are the third part of the third piece of the formed elements, okay, also called thrombocytes. They're the second most abundant of the formed elements, and they really play a key role in stopping bleeding or hemostasis, okay? Platelets really are fragments of a larger bone marrow cell, and the edges of this cell really just break off to form these cell fragments called platelets. Platelets live only about 7 days. So you can see here they're just kind of breaking off from this cell, and you can see how small they are in compared to the red blood cells. So let's talk about hemostasis a bit, and this really is kind of preventing bleeding, okay? So what happens first is we get a blood vessel that is cut, okay, and the body must react quickly to stop the flow of blood. It does so through the following sequence of events. First, we have vascular spasm, then the formation of a platelet plug, and the formation of a blood clot. So as soon as the blood vessel is injured, smooth muscle fibers in the wall of the vessel spasm, which immediately because of this vascular spasm slows the flow of blood. This response really is only temporary though, and kind of gives the other mechanisms time to activate. Secondly, we get the formation of a platelet plug. Really what happens here is that the break in the blood vessel exposes collagen fibers, you can see them there in the diagram, creates a rough spot on the vessel's normally very slick interior. And this rough spot triggers passing platelets to kind of stick to it. So it makes the platelets a little bit more sticky, okay, because there's injury there. Then they stick to this area because it's kind of rough, okay? So they kind of clump together. They also secrete a whole bunch of chemicals, and some cause the vessel to constrict further, okay, and some attract more platelets. So this platelet plug kind of forms a temporary seal in the vessel wall as we lead up to a more stable solution of a blood clot. Okay, so the formation of the blood clot is really the most stable way to stop the bleeding. Okay, blood clotting, also known as coagulation, okay, involves a series of complex chemical reactions using proteins called clotting factors. Okay, the first several reactions vary depending on whether the process has been stimulated by factors inside or outside of the blood. When the damaged blood vessels and surrounding tissues, okay, release clotting factors, a cascade of events called the extrinsic pathway begins. When the clotting factors are activated within the blood, a cascade of events called intrinsic pathway begins. The extrinsic pathway activates factor X through a single reaction. Okay? The intrinsic pathway, however, requires 4 additional reactions to activate factor X. Okay? But once factor X is activated, the formation of a blood clot follows a very common pathway. So both pathways are going to result in this blood clot and result in the formation of factor X. You just need to know that the extrinsic pathway is much more direct, going to form a blood clot in about 15 seconds, versus intrinsic is going to be a lot more steps, and it's going to take a little bit longer, 3 to 6 minutes to form a blood clot. Okay, so the common pathway ultimately leads to the production of prothrombin activator. Okay, prothrombin activator acts as a globulin called prothrombin, and it converts it to an enzyme called thrombin. Thrombin transforms the soluble plasma protein fibrinogen into fine threads of insoluble fibrin. Okay, so prothrombin activator, prothrombin, thrombin, fibrin. Okay, so that's the pathway. Okay, and then the fibrin actually forms a web at the site of injury. Red blood cells and platelets flowing through the web become ensnared, creating a clot of fibrin, blood cells, and platelets. A blood clot can effectively seal breaks in a smaller vessel. Blood clotting alone may not stop bleeding from a large vessel, though, but the body's going to try.
When a blood vessel is cut, the body must react quickly to stop the flow of blood. Immediately after an injury, smooth muscle fibers in the wall of the blood vessel spasm. This constricts the blood vessel and slows the flow of blood. Although the response is only temporary, it gives other hemostatic mechanisms time to activate. The break in the blood vessel exposes collagen fibers, creating a rough spot on the vessel's normally slick interior. This rough spot triggers changes in passing platelets, transforming them into sticky platelets. The sticky platelets accumulate at the site of the injury, forming a platelet plug. The platelets also secrete several chemicals. Some cause the vessel to constrict further, whereas others attract even more platelets. The platelet plug forms a temporary seal in the vessel wall. A more stable solution requires the formation of a clot. Blood clotting, or coagulation, involves a complex series of chemical reactions using proteins called clotting factors. The exact sequence of events used to form a clot varies, depending on whether the clotting process has been stimulated by factors inside or outside the blood. When areas outside or extrinsic to the blood, such as the damaged blood vessel or surrounding tissues, release clotting factors, a cascade of events called the extrinsic pathway begins. When factors within the blood itself, such as the platelets as they adhere to the collagen in the damaged vessel wall, activate the clotting factors, a cascade of events called the intrinsic pathway begins. The extrinsic pathway activates blood clotting factor X in single reaction. The intrinsic pathway requires 4 different reactions to activate factor X. Either way, once factor X is activated, The formation of a blood clot follows a common pathway. After factor X is activated, an enzyme called prothrombin activator is produced. Prothrombin activator acts on a globulin called prothrombin, factor II, converting it to the enzyme thrombin. Thrombin transforms the soluble plasma protein fibrinogen into fine threads of insoluble fibrin. The sticky fibrin threads form a web at the site of the injury, creating a clot of fibrin, blood cells, and platelets. A blood clot can effectively seal breaks in a smaller vessel. However, blood clotting alone may not stop a hemorrhage from a large blood vessel.
So I think that's nice because you can see the pictures, go right along with it as we're talking about it. So the other part of blood clotting is the dissolution of blood clots. So there's a couple of ways in which this happens. First part is when that fibrin creates that web, we get that nice clot there. The platelets trapped within that web contract, pulling the edges of the damaged vessel closer together. So actually part of the healing process. After the vessel is healed, the chain of reactions converts plasminogen to plasmin, and plasmin dissolves the fibrin mesh network and the clot breaks up. And this is called fibrinolysis. So it's a two-step process there, but pretty simple, pretty straightforward. We don't always want blood clots, though, especially in healthcare. We're going to talk a lot about blood clots that we don't want. We certainly have a need for blood clotting, but there are some factors that discourage blood clots. So a smooth endothelium prevents platelets from sticking. Okay, so normal circulation dilutes thrombin. Okay, when blood pools in the legs, however, though, when we sit for a long period of time or we're lying down, thrombin can accumulate and lead to clot formation. Okay, and then basophils and mast cells secrete heparin, which blocks the action of thrombin. So there's three ways that we discourage blood clots, but you can understand how each one of these three ways there might be a dysfunction too. So when someone gets a blood clot, we have to ask ourselves what went wrong, okay? And an unwanted blood clot inside of a vessel is called a thrombus, okay? And if a piece of the clot breaks off, circulates through the bloodstream, it's called an embolus. So you will need to know the difference between thrombus and embolus. Let's finish this discussion talking about blood types, okay? So everybody's probably familiar that there are blood types, okay? Call it an ABO blood typing. Okay, the surface of each red blood cell carries a protein called an antigen. Okay, there's two antigens, A and B. So people with type A blood have A antigens on their RBCs. With people with B blood have B antigens, AB blood have both, and O have neither. Okay, so that's the first step in being able to blood type somebody. Pretty straightforward. What happens next is the blood cells carry antigens, which we just talked about, and the blood plasma carries antibodies against the antigens of the other blood types. So type A blood has anti-B antibodies because we don't want to be B blood, right? We're A blood. We have A antigens on our red blood cells, so we have anti-B antibodies. Type B blood has anti-A antibodies. Type AB blood has no antibodies, okay, because they have both types of antigen. And type O blood has both anti-A and anti-B antibodies because they have no antigens on their red blood cells. So what happens when we give somebody a blood transfusion? So let's watch that animation.
The surface of red blood cells typically carries a protein called an antigen or agglutinogen. There are two types of antigens, known by the letters A and B. People with type A blood have the A antigen on their red blood cells. People with type B blood have the B antigen. People with type AB blood have both A and B antigens. People with type O blood have neither antigen. Blood plasma carries antibodies, called agglutinins, against the antigens of the other blood types. Type A blood has anti-B antibodies. Type B blood has anti-A antibodies. Type AB blood has no antibodies. Type O blood has both anti-A and anti-B antibodies. If someone receives a transfusion of another type of blood, the antibodies in that person's blood would immediately attack the red blood cells in the donor blood. For example, if someone with type A blood receives a transfusion of type B blood, the anti-B antibodies in the recipient's blood will attack the donor's red blood cells. The resulting reaction would cause the red blood cells to clump together, or agglutinate. The clumping red blood cells would block blood vessels, cutting off the flow of oxygen. The red blood cells would also burst, called hemolysis, causing them to release their hemoglobin into the bloodstream. The free hemoglobin could block tubules in the kidneys, leading to renal failure and possibly death.
So you can see why it's really important that we get people's blood typing correct before we give a blood transfusion. Now you might say, well, I always heard that type O has been referred to as a universal blood donor. Okay, remember, it contains neither antigens A or B, implying that it can be given to any recipient. This isn't true, though, because the donor's plasma contains both anti-A and anti-B antibodies. This means that the type O blood would agglutinate or clump together, okay, if we gave it to type A, type B, or type AB. Usually, though, this doesn't occur because only the red blood cells are transfused. So that's why when we give somebody O, we are just giving them the red blood cells, okay, that have neither antigen on it. And their blood, even though they have A or B and they have anti-A or anti-B antibodies, isn't going to attack that because they're not going to be getting the anti-A and anti-B antibodies from the donor blood. So that's why it's a universal donor. The other part to blood typing is the RH group. Okay, so this is when you hear of them saying RH positive or RH negative, or maybe just O positive, O negative. Okay, RH positive blood contains the RH antigen. Okay, and RH negative blood lacks that specific antigen. Blood does not normally contain anti-RH antibodies, so it's not usually a big deal. Someone with RH negative blood develops anti-RH antibodies after being exposed to an RH positive blood, either through transfusion or when an RH negative mother becomes pregnant. Okay, so there's a couple of ways in which this happens. Okay, the body naturally does this to kind of protect itself. So you can see here in this diagram that usually the first time they're exposed, it's not a big deal. But then the second time they're exposed, it is a big deal because then they have these antibodies that attack that RH antigen. Here it shows you what's happening when a mother gives birth. Okay, so she's RH negative, the baby's RH positive. The first time she has a baby, it's okay. Okay, the bloods don't mix while the baby's inside of her. After she gives birth, some of the blood does mix. So then she gets exposed to the RH positive. She develops antibodies against that. Okay. And then the next time she has these RH antibodies, the baby has RH positive. Again, those antibodies are going to attack the baby. So that's why it's really important that we know whether somebody is RH positive or RH negative.
Besides being classified according to A, B, O type, blood is also classified as being Rh positive or Rh negative. Rh positive blood contains the Rh antigen. Rh negative blood lacks this specific antigen. Blood does not normally contain anti-Rh antibodies. However, it is possible for someone with Rh negative blood to develop anti-Rh antibodies. There are two ways this can occur. The first way is when someone with Rh negative blood receives a transfusion of Rh positive blood. The recipient's body interprets the Rh antigen as something foreign and develops antibodies against the Rh antigen to protect itself. If the recipient has a subsequent infusion of Rh positive blood, The anti-Rh antibodies that form during the first transfusion attack the Rh antigen in the donor blood, causing agglutination. The second way someone can develop anti-Rh antibodies is when an Rh-negative mother becomes pregnant with an Rh-positive fetus. Because maternal and fetal blood does not mix, the first pregnancy with an Rh-positive fetus will proceed normally. During delivery or miscarriage, the fetus's blood often mixes with that of the mother, thus introducing Rh antigens into the mother's bloodstream. The mother's body responds by forming anti-Rh antibodies against this foreign substance. If the mother later becomes pregnant with another Rh-positive baby, the anti-Rh antibodies can pass through the placenta even if red blood cells cannot. When they do, they will attack the fetal red blood cells, causing agglutination and hemolysis. This will produce severe anemia in the infant, called erythroblastosis fetalis.
So what I want you to know is the difference between that people have Rh positive and Rh negative, but you don't need to know all the different steps of how this occurs. I just wanted to show you it. What we do to prevent this is when we test the mother and the mother is Rh negative and we know she's having the Rh positive baby, she's treated with RhoGAM, which is a way that we prevent the body from forming these anti-Rh antibodies. Okay, so we do have a way to prevent this and treat this.